morning, one and all. Good morning, honored guests and esteemed delegates. It's my great pleasure to welcome you all to the International Conference on Radiation Research, Impact on Human Health and Environment, being held from November 22nd to 2024 at the All India Institute of Medical Sciences, Patna, and Sri Sai Institute of Medical Sciences, Hajipur, under the aegis of the Society for Radiation Research, marks the fourth biennial meeting of the SRR and is a significant occasion for the radiation research community. As we gather together, explore the cutting-edge advancements in our field, I would like to take a moment to acknowledge the Society for Radiation Research. SRR has been instrumental in fostering a multidisciplinary approach to radiation science, uniting researchers, clinicians, and industry leaders to address clinical challenges and drive innovation. Its commitment to promoting collaboration and excellence makes it a cornerstone progress in radiation research, setting the stage for impactful discussions like the ones we will have today. The Society for Radiation Research has been a pioneering platform in fostering collaboration and innovation in the field of radiation research. Since its inception, SRR has been dedicated to advancing scientific knowledge, supporting young researchers, and promoting interdisciplinary approaches to understand and mitigate the effects of radiation on human health and environment. This conference also celebrates 10 years of SRR's remarkable contributions to this critical field, making this gathering all the more special. With a diverse lineup of topics, esteemed speakers, and enthusiastic participants from India and abroad, ICRR HHE 2024 promises to be an engaging and insightful journey. On behalf of the organizing committee and SRR, I extend a warm welcome to all the participants. Let us look forward to a fruitful discussion and meaningful collaborations during these 3 days. Thank you all. We now move on to one of the most esteemed sessions of this conference, the SRR A.R. Gopal Iyengar Memorial Oration Lecture. This prestigious lecture commemorates the extraordinary contributions of Professor A.R. Gopal Iyengar, a pioneer in radiation research, whose legacy continues to inspire scientific progress. It is a tribute to his vision and dedication to advancing the field. It is now my pleasure to invite our chairpersons, Dr. K.P. Mishra, Founder President SRR, former Vice Chancellor Nehru Gram Bharati University, Prayagraj, former Head RBN HSD, BARC Mumbai, and together with Dr. K.P. Mishra, we also welcome Dr. Akhileshwari Nath, former Head Research Center Mahavir Cancer Sansthan, Patna, to chair this session. Please join me in welcoming them to the stage. I now invite the chairpersons to take over the session and introduce the speaker.
Yes, I'm audible. Okay. Welcome and good morning to the very first session of this international conference on radiation research organized by Society of Radiation Research of India. Ladies and gentlemen, this session is the most prestigious lecture in the honor of Dr. A.R. Gopal Iyengar, a famous scientist from Bhabha Atomic Research Center and known the world over in the field of radiobiology, cell biology, and mutation research. Dr. A.R. Gopal Iyengar was born in early 20th century in 1909, and he was director of Bhabha Atomic Research Center for 2 decades. And all the research in radiobiology was conceived, planned, and executed by the late Dr. A.R. Gopal Iyengar. We are all product of, at Bhabha Atomic Research Center, we are all product of what he conceived in 1960s at that center. Dr. Gopal Iyengar was a pioneering research scientist in the field of radiobiology and also in allied sciences. He was very particular to promote basic radiation research, at the same time applications of radiation in human health, agriculture, medicine, and so on. So we are very, very grateful to this scientist who we are going to honor. And in his honor, we have invited Professor George Iliakis, Director of Radiobiology Institute at Essen, Germany. May I invite Dr. Iliakis to the stage, please? Yes, please, and welcome him with a applause. Dr. A.R. Gopal Iyengar was basically a nuclear medicine scientist to begin with, a cytologist, and he was invited by Dr. Bhabha. He was trained in United States in Berkeley Institute in nuclear medicine. Okay, most welcome. Please come here. Most welcome. And he was invited by Dr. Bhabha to head the then cell biology division at Bhabha Atomic Research Center. And he was the recipient of the civilian Government of India Award, Padma Shri, in 1967. He also received the Anne Winter Corby Award from Toronto University in 1941. And he received H.J. Bhabha Prize, which was given to outstanding scientists in 1948. He was a fellow and president of Indian National Science Academy and fellow of Indian Academy of Sciences, visiting scientist in many institutes of Germany, United States, and Japan and UK. He published many papers. He was vice president of IUPAB. He was also president of International Society of Cell Biology, established many institutes and universities in India. So ladies and gentlemen, in the honor of the scientist who was originator of radiobiology research and applications in India and played a very decisive role in development of atomic energy program of this country in 1950s. In his honor, the Society of Radiation Research has instituted this prestigious lecture, and this lecture was given by very prestigious people, very well-known scientists of the world. And I recall the first lecture was given by Professor Atkinson from Radiobiology Institute, Munich, in 2016. And the second lecture was given by the Armed Forces Radiobiology Institute director, Dr. Huff. And then Dr. Gale gave the lecture in 2022 at Mumbai at Bhabha Atomic Research Center. And the fourth lecture is going to be delivered by Professor George Iliakis from Germany. I have a privilege and feel honored to invite you to give this lecture, that you were kind enough to have accepted our invitation. Let me just mention to you that George has been a scientist who needs no introduction to radiation research scientists the world over. A very accomplished scientist, very dynamic researcher, and a leader. He was the director of the Clinical Radiobiology Institute at Essen in Germany, and he is now president of International Association of Radiation Research. He received back an Alexander Award of European Society, Ulrich Hagen Award, and 20th Radiation Research Young Investigator Award of Radiation Research Society. Ladies and gentlemen, Dr. Iliakis is a leader of world class with novelty of research in the area of DNA damage response and emphasis on DNA double strand break repair. We all feel honored when George Iliakis is with us in the international conferences. So I invite Dr. George Iliakis to deliver this prestigious lecture of Dr. A.R. Gopalayanar, and request you to welcome him by applauding and welcoming him. Thank you. So, George, floor is yours now.
Well, I have to say that I'm deeply humbled and also deeply honored to be given this very prestigious lecture. And I would like to thank Dr. Mishra and all the organizers, for that matter, for the distinct honor that they gave me by asking me to give this lecture. Let me get to the first slide. Okay. I have to tell you that one of the things that always puzzle me when I think about what to do next in our field is, what do we already know? And although we use radiation and we apply the word radiation, we try to find ways to protect ourselves from radiation or to apply it in all these different disciplines, it is surprising and in some ways also a little bit scary how little we know about it. And what I will try to do with you today, I will start from the very beginnings of what actually radiation is, and I will show you that although it's something that it should be very easy to understand, at the end it becomes very complex, and it actually touches the very being of of our genomes and of our genomic stability. And that's why we connect it also so much with cancer. Before I even start, I would like to say that, of course, what I'm going to talk to you about today, I haven't done myself. I have very excellent colleagues that actually throughout the years, I'm now in the field over 40 years, and it is only through my collaborators that I have been able to achieve what we have achieved. So great thanks goes to all of them. Ashish Soni is also still with me and also around in this meeting. Also, what is also very fair to say is that it would have been impossible to do what we did if we didn't have support. And what you see here as all the agencies, which throughout the years, really in an excellent way and trustfully, they gave us the money we need to do this kind of research. So thank, first of all, to all of them. Now, let's go into the basics of radiation and radiation biology. And I don't need to tell you that radiation is used in medicine, energy production, diagnostics. It is all over the place because it's terrestrial radiation and also it's cosmic radiation that we care about now. The space age that now actually starts really, it becomes a very important issue, but we know also that there is something we have to worry about radiation. And this is because we know, beyond any doubt, that it does generate actually genomic instability and that it actually leads to elevated cancer risk. So we have the duty as radiation biologists first to understand what radiation does to improve its medical use and also to understand what radiation does so that we can protect humans actually from the adverse effects of radiation. And we heard last night an excellent session where those topics were addressed in a very specific and actually concrete way. Now, let's go back. What is actually ionizing radiation? And the surprising thing actually is that ionizing radiation is exactly what the word says, which is energy that can ionize. Nothing more than that. But one thing that we tend to forget is that when we talk about ionization, what we really mean for the molecule or the atom that has been ionized is actually that it lost an electron. But go back to your chemistry. The chemistry classes, and you remember that loss of an electron is actually an oxidation reaction. Okay? So in chemistry, it's just an oxidation reaction that actually radiation is doing. And the question is, but how come that when we talk about hydrogen peroxide, a typical oxidizing agent, we worry so much, we worry very little about, but when we talk about radiation, we worry more. And actually, the first thing I want you to take with you today is that the only reason ionizing radiation is more than hydrogen peroxide is because the ionization events it generates, it has a little bit physics here, and I don't have the time to go into it, but some of the energy ionizing radiation deposits, it is deposited not in single ionizing events or oxidizing events as we hydrogen peroxide, but actually in clusters of oxidization events. And it is actually this clustering that generates the difference between hydrogen peroxide and ionizing radiation. Now, this is X-rays, what we used to call sparsely ionizing radiation. Now, again, a very hot topic in radiation biology is like when we talk about particle radiation. particle radiation, like for example, an alpha particle, then you have an even stronger clustering. And what this clustering does, if it meets the DNA, is that it will damage the DNA beyond what a simple oxidizing event will actually do. And that is actually the entire difference between the two events. It's only levels of clustering that matter. But now let's go one step further to see the effect. And this is where the result is becoming absolutely bluffing. Okay? And this is because, look, hydrogen peroxide, now this is survival. We see how we kill the cells as a function of those. And what you see is hydrogen peroxide will barely do anything to the cells that they cannot deal with. But when it comes to X-rays, you see a tremendous amount of killing. And what is even more important is, as you increase the LAT of radiation, like you make the radiation more densely ionizing, then you get more and more effect. And you see what you get when you get the alpha particles that you see here. So this densely ionizing, and you get this tremendous response, then a little less, and you get this response. See how different it is. And hydrogen peroxide, especially ionizing here and there, and then you get the response that you see there. So it's a fantastic effect, and make sure that you keep this difference that I show you here in terms of difference in survival and so on. Every time you see effects of radiation and what the radioprotectors or radiosensitizers are doing, repair inhibitors, or for that matter. Because if you compare the effects that you see here with the effects that you see there, you see almost nothing will overshadow this. And needless to say also that this effect is also generating the need to define quantities like the RBE, on which actually the effects of radiation of different types of radiation are measured. And this is very intensively used, of course, in radiation protection. Okay, now, I told you that radiation is effective because of the clustering. And who cares about clustering? Why should we care about clustering of ionizations? Now we leave the physics and go into biology. And now we look the molecule that we think is at the beginning and the end of most radiation effects, and this is the DNA. And 50, 70 years now, we have experiments that actually suggest that the most important target to radiation is actually the DNA itself. And second, that the generation of a type of lesion, which we call the DNA Dobson breaks, which is nothing else that you actually break the molecule. No surprise there, because if you have this clustering, okay, and you have this clustered ionizations, you can break the DNA so close together so that the two molecules, despite the fact that they have two strands, okay, they can break. And you have a Dobson break. So, and this is the base of radiation biology. And actually, if you ask what I've done in my 40 years of radiation biology life, that's it. All I care about is DNA Dobson breaks. Okay, if you look my caplates, it's DSB 9010. Okay, so it's all I'm thinking about and all that I have done throughout the years in the field of radiation. And this, what we try to do, or what we know about this, I will tell you in some ways in the following slides. But what I would like to point out today, that although we start now the biological trip into radiation effects from the Dobson break, even there now the fields are getting complex, because what you see is Dobson breaks can be generated by restriction endonucleases. Biology generates Dobson breaks to help itself in a certain ways, okay? But these Dobson breaks are just the cut of the DNA strand in a very clean way, and the cell can put it together, no problem at all. When radiation generates the Dobson break, like the way that I showed you just a few seconds ago, we actually change molecules that part of the DNA. We change nucleotides. And of course, that generates trouble for the cell and makes the reconstitution of the DNA more complex. In the field of radiation biology, the common explanation of the high LT effect is that the complex lesions are generated, which are actually causing not only the Dobson break, but actually additional changes close to those breaks, and that actually generates what we have as a high LT effect that I showed you the slide before. However, in our view, that doesn't explain what we see and the level we explain. So we have gone into long trip, try to classify the different Dobson breaks. So it's not a Dobson break, but there are different types of Dobson breaks. And we think that these different types have different effects in biology. And we sort of postulated that actually the complex lesion that generates the high LT effects is not the complex lesion that adds to the break also some additional base damage, but actually clusters of Dobson breaks. And this, we are not the first to do that. There is a lot of research along those lines, and what you actually see as classical explanation, clusters of Dobson breaks will let DNA move out of the genome, get lost. And of course, that's not a good thing to happen. And also, we have plenty of models of and experiments with pulse field electrophoresis that actually show that we do get small pieces of DNA when we see how is the DNA broken using pulse field gel electrophoresis. And also, if you do cytogenetics, you see plenty of single, of small pieces of DNA of chromosomes that suggest that indeed clusters of Dobson breaks or pairs of Dobson breaks are actually generated. This will be the defining part of one of the sessions that I'm going to, of the effects that I'm going to study with you today, and I'll tell you what we're doing. So we have the physical effects, we meet the DNA, and we now think about the complex lesion as a cluster of Dobson breaks. Now, we have different types of Dobson breaks, and now the question is what the cell is doing with them. How is the cell responding to Dobson breaks? And again, 30 years of work allow us now to postulate or to at least show that once you get a Dobson break, then the first thing that is going to happen is the ends will be bound by a molecule that is called Q. There is nothing in the cell that would bind DNA ends stronger than Q. And as a result, it's difficult to understand how anything else but the pathway that you see here is going to be operational, which is like after Q binds, then you have additional molecules that go on, including a PKCS, very important molecule, ligase 4, etc., to end up in an enjoining reaction that we call classical non-homologous enjoining. So that's something that one needs to keep in mind. However, the last 20 years has become very clear that despite the binding of Q at the DNA ends, some Dobson breaks will actually be processed and in a reaction that we call resection. And this resection is actually going to lead to other pathways that are going to work. And the first one, and probably the most important one, is the homologous recombination repair, with RAD51 playing a very important role here, but also with single-strand annealing also playing a role, and most importantly also the alternative enjoining. Now, we end up with 4 pathways to repair a Dobson break, which is actually a lesion that we know we have to worry about. And this is good news because Dobson break is a bad thing to happen in a cell, and the cell is equipped very well with different repair pathways to repair the Dobson breaks. Where it becomes puzzling and generates all sorts of philosophical and research-wise question is if you look carefully at the properties of these repair pathways, and you see that the classical enjoining pathway is actually error-prone, it's fast, and cell cycle independent. And in the homologous recombination is error-free, but is slow and only works in G2 phase of the cell cycle, S and G2 phase of the cell cycle. And the single-strand annealing is actually error-prone, slow, and cell cycle dependent. And finally, the alternative enjoining is error-prone, slow, and also cell cycle dependent. Now, the alternative enjoining has been a pathway that has occupied 10 years of our work in the lab. And actually, where we start publishing on that field back in the year 2000, and we published a lot of different papers with a lot of different characteristics and different parameters that are involved and different proteins that are required and so on. And actually, we were publishing those papers, but they didn't find that much attention, really. And it was actually only that the importance of the alternative enjoining pathway became clear when Fred Old and David Roth, they published 2 Nature papers that actually show that alternative enjoining is actually involved in 2 key pathways of the immune system. You see how different areas of investigation actually meeting sometimes in surprising ways. And this is actually VDJ recombination, and this is class switch recombination. And once you get those pathways working, you will not have a functional immune system. So, and now, if I take 1 second to summarize what I told you properties of radiation that generate Dobson breaks, different types of Dobson breaks, and also showed you that non-homologous enjoining is actually the pathway you would expect to work. But there was this also other pathway, homologous recombination, and now the question is, when the cell needs to decide what to use, what parameter does it use, what is gauging in order to make the decision, and here there are a lot of different alternate ways to explain what it happens, and I'm going to show you now what is our work, because needless to say that we spent a lot of time working on alternative enjoining pathway, but that was not what we were really interested in. We were interested in how cells repair Dobson breaks overall, like the logic that is behind what repair of Dobson breaks actually involve. So now I'm coming to the topics of the talk, and what I'm going to discuss with you today is how is the selection between non-homologous enjoining and homologous recombination repair. Now remember, classical non-homologous enjoining will be error-prone. Homologous recombination repair will be error-free. Okay? So this is a very important decision. Okay? Whether you take the wrong or the right pathway will have tremendous consequences for the genome of the irradiated cell. And also, the last thing that I'm going to show you today is some very recent results. Actually, the paper will come out in radiation research in the next few months, I hope. And basically, I want to show you that something that every model of radiation action was considered as a given, that means a surviving cell, a cell surviving the radiation insult has the genome of the pre-irradiated cell is actually not the case. And actually, genomes of surviving cells of irradiated genomes have tons of changes to worry about, and that is go at the heart of course, of carcinogenesis or radiation carcinogenesis. Now, let's start now with the results, and I will try to go quickly through those. First of all, non-homologous enjoining, homologous recombination repair. That's the basic question, and many people tell you it's stochastic. Okay? And what I show you now, evidence that this is not the case. So what did we do? Of course, we took cells in G2 because these are the cells that do actually homologous recombination. Other cells cannot. So, and then we study in G2 phase cells, we studied actually the formation and repair of Dapsone breaks by measuring gamma H3X, what you see here, standard experiments. There are millions of papers that are published in this topic, and you get these standard curves. And then we go to the protein that I already mentioned to you, namely RAD51, which is a protein that is very specific actually for homologous recombination. So when we see a RAD51 foci, we can infer there is homologous recombination working there. And now it's not rocket science to say that if we now take the total breaks, which is gamma H3X foci, and the RAD51 foci, and generate the ratio, it will reflect the ratio of Dapsone breaks that are processed with homologous recombination. And actually, the final result is what is stunning. Because if you look at this ratio at very, very low doses of radiation, then you see that you have 60% RAD51 to gamma H3X foci, which means a Dapsone break is induced, and 60% of the time, homologous recombination will work. And this is G2 phase, remember that. We go to a dose of only 2 gray. For radiation oncologists, it's a low dose. For radiation protection people, it's a huge dose. But what you see, we are now down to almost 20%, and you go one step further to really high dose, 60 gray, and you are down to 1 or 5%. So you see, from 60% to practically nothing. This conundrum is what took our work for 20 years, because if you do experiments here, and we were doing these experiments here, you say there is no homologous recombination. Homologous recombination doesn't play a role. And there is tons of data that show this black on white. And then there are other experiments, like, for example, if you have the reporters, GFP reporters, that show that this is actually not the case, because for reporters, when you have only one single Dapsone break that you ask the cell to repair, then you see that you have practically from 50 to 0, exactly what I showed you in the slides before. So, what now this tells you is what pathway you use depends on the number of Dapsone breaks that you have in the genome. So that is very important to keep in mind, both for the clinic and for radiation protection. Now we go to the second question, and this is a question of complexity. And I told you, we don't buy that complex lesions are lesions that have 2 single-strand breaks and some base damage close by. I'm not saying they're not more consequential than the simple clean Dapsone breaks, but what I'm saying is they do not explain the high LET effect by themselves. They contribute, but they don't contribute 100%, and we actually think that what we have as a major contributor is classes of Dapsone breaks. Now, you can talk a lot, you can make models, you can do a lot of different experiments and so on, and you are besides the point always, because it's actually conjectures. We wanted to see whether we managed to generate real evidence that clusters of double breaks actually are more of a problem than when you have single double breaks. So we thought, let's do real molecular biology. So everybody's using ASU1 to generate double breaks in the cell and then do some very cool type of experiments to find out how cells respond to them. We thought, let's do the following. We take ISU1 and we generate singlets, doublets in different distance, doublets with different ends. That also is part of the complexity. We even went to even further and saw, okay, let's even do quadruplets and see what happens. So that's the first thing we do. We change complexity. The second thing we did is we used a transposon system that allowed us to put these constructs, these are actually constructs that we can transfect the cells with, and they get integrated in the genome. And then we can have multiple integrations. Okay? We were not interested in single integration that every molecular biologist that uses SU1 tries to do. We wanted to have a lot so that we somehow mimic the effects of ionizing radiation on, in those plasmid transfected cells. And when we did that, and to our great surprise but pleasure, what we found was that the more complex the double break was, let's say 2 or quadruplets and so on, compared to the singlets, see one double break, two different ends. All you need to do is look at the number of colonies, and all you need also to do is look at the survival. The more complex the double break you put into the cell, the easier it is to kill the cell. Rings a bell? I showed you the curves. With alpha particles, how much more killing you are getting by having this complex, which we didn't know what it was. But high LET is extremely more effective than low LET. And you see it also in the number of translocations that you're getting. These are precursors of cancer, and therefore it's very important that they are generated with this defined double breaks, I would say. So that was very encouraging, and we thought, okay, that's something in the right way, and we thought, but can we actually confirm this result using something alternative? And what we did is we found a good way to repeat this experiment using the CRISPR-Cas9 model system that I'm sure you all know about. And we now said, let's generate using a particular genome site, and this is the site of the HPRT gene, which you see here with texons. And we selected exon 3, and we decided, we designed guide RNAs. This is my time. Can I have another 7 minutes? Okay. And we decided to generate double breaks in different locations in the genome, and to see what actually happens. We use the classical HPRT system for mutation induction, and we looked through, after transfection of the Cas9 and the guide RNA vectors, to generate the double breaks that I showed you before, to what kind of mutants we are getting. And this is the answer, and it reminds actually in the answer that I showed you before, because you see, if you have nothing, you get no mutants. If you have a single double break, you generate mutants. But look, if you add 3, 4, 5, 6 different double breaks, so if you increase the complexity, then you get a lot more. So this is exactly what we showed before for the entire genome. And in this slide actually shows you that there are distance also that play a very important role. So if you say you take 2 and 6 as opposed to 3 and 5, let's say 2 and 6. Far away, 4 and 5, close by. You see, if you are close by, you generate a lot of mutants, but if you go far away, it's like the two Dobson breaks do not combine to generate a complex lesion and you get no mutations. So this is a very convincing way that the complexity depends on the distance between the different Dobson breaks, and also that it has the PI3 kinase family of kinases. It's actually very much involved in this type of experiments. So now I complete also the second. Now I explain to you that there is a good chance that the complex lesions are Dobson break pairs or more clusters. So again clustering, but not of individual ionizations, but of Dobson breaks. And now come to the last point, which is like, what is radiation doing actually to cells that survive? That was very intriguing to ask a question, because as I told you, I grew up in radiation biology thinking that the surviving cell has restored its genome completely. Okay? And what I will show you now in the next 3-4 slides, is that this is not the case. How we did that? We irradiated cells, and actually we took H6H3 cells, which are normal cells. They're not tumor cells. We did it in purpose because tumor cells are genomically unstable, generate additional parameters and problems to this kind of experiment. So, and then we irradiated them different doses, and then we let them form colonies. So a colony is a surviving cell, of course. And then we pick the surviving colonies, each one individually, and we put it in a separate dish, and we let it expand to form like a culture. So we generated cultures that originate from a single irradiated but surviving cell. And then we do next generation sequencing, and we look at the genome of those cells. And to our surprise, this is what we found. The genomes of irradiated cells, surviving irradiated cells, their deletions, duplications, inversions, and translocations. This is what we looked for, and what you see is we find all of them. These are surviving cells, and look how many changes they have in their genomes. And this you can see also, this is now from chromosome 6 and chromosome 12, regions of the chromosome, and you see all the sequences that actually are lost. So and this is the beauty of this next generation sequencing, because you actually see what is happening in the genome in extreme detail. And you can go on also and write this kind of presentations that actually you will see very frequently in cancer formulations of all characterization of cancer cells, and what they depict here, you see translocations are like this, deletions are the red, duplications are the yellow, and so on. And what you see is these are our different clones, and you see all of them. We didn't pick one single clone that will be clean, that will have actually the original genome. And we could confirm many of those results doing cytogenetics, because some people say, if I don't see it, I don't believe it. So that's why we were very happy that we could see those things. And that actually brings me to the end. And thank you for the extra time. And I will tell you now where we are. We are working for a long time, but we're still not done. Actually, to me, it seems like we're just beginning to really understand what is going on. But you see the beauty of going into the very, very basics. I started you from the physics of radiation and got you into the lesions that are the Dobson breaks and in the way they're processed. So what do you think is what we will be looking at next? Is of course something that it's at the heart of what we try to ask is classical versus homologous recombination. I showed you something about the involvement and the dose dependence. It's extremely dose dependent. But the question is why? And if you think a little bit about this question, you will think there are 10 other questions that you will ask based on this simple finding. So that is something that we're going to work very intensively in the next few years. And we want to characterize that the parameters that affect the selection based on the load. And we want to study the role of chromatin in these processes. And in the end, we think that the cell is like an intellectual system that applies necessity. If it's a complex topson break, it has to do something. If it's a simple topson break, it has to do something else. But it has also logic, which means that if there is an error-free pathway, it would be stupid not to use it unless some other parameters generate this sort of necessity and stop it. So that's also a key question in what we're doing. And with this, you see the group again. And actually, I moved now into the Department of Radiation Oncology, and Martin Stuschke is the chairman of the department, and we're grateful that he actually took the radiation biology when the dean decided with my official retirement that it's time now to stop having too much radiation biology on campus. But he gave us the opportunity to continue this work, and we are very thankful and appreciative for him. So that was it. Thank you very much. And if you have questions, I would be happy to try to address them.
Thank you, Dr. Ilyas. Thank you very much for coming from such a long distance for this conference. I know the delegates have number of questions in their mind, but in oration lecture, we should not ask questions. You can discuss it in lunch. Thank you, Doctor, for very good informative lecture. You have explained in a very simple way about the IR radiation, about the mutation occurred, and how the damage is repaired by different pathways, even by backup pathways. Thank you, Doctor. Thank you very much.
Thank you.
That was a wonderful session. Thanks for sharing your insights, sir.
Okay.
I now call upon stage our honorable chairpersons, Dr. K.P. Mishra and Dr. Akileshwari Nath, to felicitate Dr. George Iliakis. I would like to ask Dr. Pritanjali Singh to felicitate Dr. KP Mishra.
Excellent, excellent. I return to my seat now? Or?
Yeah, this thing, yeah.
I would like to ask Dr. Anita Kumari to felicitate Dr. Akhileshwari Nath.
It's an alluring day, and I extend my warm welcome to all the dignitaries gathered here for this influential event. Next on board, we have a presidential session, Radiation Oncology and Hypothermia. I take the honor to invite the chairpersons, Dr. Ramesh S. Bilimaga, President of the Society for Radiation Research, SRR, and a senior consultant and professor emeritus of radiation oncology at HCG hospitals in Bangalore. Dr. Rajesh Kumar Singh, Professor and Head, State Cancer Institute, IGIMS, Patna. Dr. Pankaj Kumar, Assistant Professor, Nuclear Medicine, Aiims, Patna.
Good morning. Maybe all of you are wondering, there's one more talk which is supposed to be given by Avinash Pandey is not here. But this talk will be given soon after this particular presidential session is over. So you will not be disappointed at all. The present session has 5 lectures in this, and I have 2 co-chairmen to assist me. And we will call the first talk by Hans Gries. Yeah, is it? Yes. Can you come out to the dais, please? He is from the University of Amsterdam, Netherlands, and his presentation is IL-1 and clinical and preclinical evidence and for efficacy of concurrent hyperthermia and radiotherapy. And he has been working in this field for quite some time. In any of the national, international conference, if you see for the hypothermia, and professor's name is always there, and his lecture is always there. He is a very, very dedicated worker and plus a researcher and a good speaker too. Welcome, Lance. Go ahead.
Thank you very much for the introduction and also for the invitation to enlighten you about the benefits of combining hypothermia with radiotherapy. So first of all, for those who are not familiar with that, hypothermia in this framework is raising the temperature of the tumor to 40 to 43 degrees for 1 hour, and always to be combined with either radiotherapy or chemotherapy or even immune therapy. And the goal is to achieve doing so in tumor selective sensitization, which we can achieve if we keep the temperature below 44 degrees. This treatment is actually already performed for a great deal of different tumor sites and with very good results when combined, for instance, with radiotherapy, adding about 15% to the response rate. Now, hypothermia is actually in principle a wide range of different applications, going from very low to very high temperatures, where the high range is used as a direct thermal ablation technique. And not just the temperature range, but also the duration of treatment plays a major role in determining whether the effect is direct ablation or a sensitizing effect. So what is the biological rationale for doing this? And I will focus mainly on the rationale as conceived also in the clinical application. Because if we look at the many effects of hypothermia, you can imagine that even a modest temperature rise is causing a host of different effects in the tumor cell and also in normal cells. From these effects, I will focus on those that we know play a major role in the clinical application. One of these effects is the elimination of hypoxic tumor cells. We know that radiotherapy in itself is less effective in a hypoxic tumor microenvironment, but also that hypothermia on its own is already capable of eliminating these hypoxic tumor cells. And cells are particularly sensitive to hypothermia when they are under chronically hypoxic conditions. So the longer the hypoxia is in place, the more effective the hypothermia is. And we also know that there is a strong dose-effect relationship. So the higher the temperature, the more effective this effect. A second effect, and in that sense it's good to be talking after the presentation, the very interesting presentation by George Iliakis, is that hypothermia can inhibit DNA damage repair. And we know that already for quite a long time. If we look at a typical response of tumor cells to radiation, we can see that at low dose rates we have only a partial effect of the radiotherapy, and that's because part of the DNA damage can get repaired. And if we then add hypothermia, in this case simultaneously with the radiotherapy, we can see that this repair effect is completely eliminated. And this is because this high dose of hypothermia can block DNA repair. If we look at the different repair mechanisms, and all of these were already mentioned in the previous presentation, all of these are actually affected by raising the tumor temperature, although some effects already at lower temperatures than others. For instance, homologous recombination, also discussed in the previous presentation, depends on a host of different repair proteins, and BRCA2 gets depleted temporarily by hypothermia, and that gives a time window to exploit if you combine radiotherapy and hypothermia. This happens to the level that if we look at the difference between the effect of low lead versus high lead radiation, as depicted in this quite old slide, we can see that if we then add hypothermia to the low lead radiation, effectively we achieve high lead radiation effects. These two effects are important. Another relevant effect is that if you locally heat a tumor, the body will respond by opening up the blood vessels in that region in order to counteract the temperature rise. And this effect can be actually quite strong depending on the tissue type. So skin and muscle tissue will respond very heavily, but also most of the tumors will also experience more elevated perfusion levels. And we also know that this can result in reoxygenation of the tumor. And the results shown here are actually achieved in PET animals treated for spontaneous tumors with the combination of hypothermia and radiotherapy. And the results show that the temperatures in a range of about 41 to 43 degrees can result in a lasting effect on the oxygenation, because also 24 hours after the hypothermia treatment we still see an elevated oxygenation. And if the temperatures get higher than that, then actually that results in the opposite effect. So temperatures between 41 and 43 are optimal for this. And this also appears to be associated with a better tumor response. And this is quite relevant because we know that tumor hypoxia can result in increased resistance to therapy and also to a higher degree of malignancy. And of all the approaches ever attempted to counteract hypoxia, hypothermia is actually the best proven method to achieve reoxygenation of a tumor. And if successful, we also know that we do have not just an improved local response to treatment, but also less metastasis afterwards. Then also hypothermia has immunogenic effects. So one effect that was already well known is the activation of heat shock proteins, but also differences in expression on the outer surface of the cell. So the heat shock proteins themselves, exosomes creation, direct effects on the immune cells, the tumor vasculature, and that has led also to the initiation of a number of clinical trials to also combine hypothermia with immune therapy in a multimodality regime, so also with radiation therapy or chemotherapy. These are presently still under investigation. One very relevant effect in the clinical application of hypothermia is also to take into account thermal tolerance, and that is that if we give a hypothermia session, that the tumor will be less responsive to hypothermia for a few days after the treatment. And that was demonstrated very adequately by giving a single hypothermia treatment, then followed 1, 2, or 5 days later by a combined treatment with radiotherapy. And that showed that initially just that combined treatment would have resulted in almost complete response. But if you have this primer treatment with hypothermia alone, then after that, for a few days, the combined treatment is less effective. So most of the clinical protocols are actually giving hypothermia once or twice a week with 3-day intervals. So if we summarize these mechanisms as shown just before, we see that they are additive and complementary in the sense that hypothermia is better at eradicating the hypoxic tumor cell fraction as opposed to the effect of radiotherapy. But there is also a synergistic effect. So radiotherapy induces DNA damage, and hypothermia can prevent the repair of that damage. But also an indirect synergistic effect, so if you enhance the local perfusion and re-oxygenate the tumor, then also obviously the radiotherapy becomes more effective. And parts of these effects are clearly tumor selective, so that also makes this combination treatment very logical. The challenge is that all of these effects I've just sketched have their own individual dose-response relationship and also their individual, let's say, optimal combination in terms of sequence with radiotherapy. So the optimal temperature also depends on this mechanism, because for enhanced perfusion you need not very high temperatures, but also not too high, not exceeding, let's say, 44, 45 degrees. For the direct cell kill, you need already slightly higher temperatures, but also temperatures quite achievable in a clinical setting to inhibit DNA repair damage. Temperatures over 41 degrees are needed, and so if you want to really benefit from all of these effects, you should aim at temperatures between 41 and 43 degrees centigrade. And we also know that the dose-effect relationship is quite steep. So any almost tenth of a degree higher, let's say half a degree higher, is already more beneficial, in the sense that if you get 0.5 degree higher temperature in this hypothermia treatment, then actually the effective dose improvement is a factor 2.5. So that's really very important to bear that in mind. Which also means that during treatment you need to monitor and verify the temperature you achieve. And this is also shown in clinical results. In this case, the results for the combination of radiotherapy and hypothermia for locally advanced cervical cancer. So the higher the temperature achieved, in this case a temperature rise between 2 and 4 degrees over 37 degrees, the better the temperature rise, the better the effect also. We also looked at the results for the combination with re-irradiation and hypothermia for recurrent breast cancer patients. This is in a post-operative setting. So these patients, when they are treated with the combination treatment, only have microscopic disease at that moment. And we found that with a cutoff point of about 40.5 degrees, if you have a temperature below that, you have a significantly lower response rate than if you achieve higher temperatures. So the difference is about 20% in this case. If we then try to analyze that by looking at all the patients in this cohort at the temperature achieved for a continuous hypothermia dose-effect relationship, you can see here that if you have a really low temperature, you achieve about 50% tumor control with radiotherapy alone in this case. And if you manage to get the temperature given here in cumulative effective minutes at 43 degrees in the best session, if that is over 5 minutes in the best session, you achieve beyond 80-90% tumor control. So way better. And that actually means that the temperature you need to achieve is about 40.5 degrees in the entire tumor and over 41 degrees as a median temperature. So that's a marked difference with the effect of radiotherapy alone. And you might think, what about toxicity? All of the toxicity we can see is due to the radiotherapy, the cumulative radiotherapy effect. And it doesn't make a difference whether we have a low or a high temperature in the tumor in that respect. The sequence and time interval is also quite important. That was studied in a very elegant study in implanted tumors in the hind leg of a mouse, which allowed to look at the effect of the time difference between hypothermia and the radiotherapy, plotted here on the horizontal axis, and also the sequence, so either hypothermia before radiotherapy or the other way around. And what you see here is that if you give the treatment simultaneously, as shown in the middle, you get a very good thermal enhancement ratio, so 2.5, which means 40 gray is the same effect with hypothermia is the same effect as 100 gray alone, but it's both for the normal and the tumor tissue. You need to give the treatment in a sequence either before or after in order to spare the normal tissue. And actually, if you do the hypothermia after radiotherapy, you can even achieve protection of the normal tissue with a longer time interval. And this is very simple to interpret. The effect 24 hours before or after is the independent effect of the hypothermia, and if you shrink the time interval, then you get the synergistic effect. And for normal tissue, of course, no enhancement, only when you get very short time intervals. And we have also validated that in our clinical cohort, where we saw that if we had a short time interval between radiotherapy and hypothermia, we had way less recurrences than for a longer time interval, where the cutoff point in this case was 80 minutes. And this also translated in worse overall survival for the longer time interval. And toxicity did not differ. So the optimal timing really means that part is independent and part is synergistic with better effect for short time intervals. So to exploit the full effect, you need to aim for a short time interval, but not too short. And the same we can see for the combination with chemotherapy, which for the combination with chemoradiation or chemotherapy alone is also relevant. The same type of effects, with an added effect that at higher temperatures the cell membrane becomes more fluid, and also the chemotherapy can pass into the cell more easily. And many of the chemotherapy drugs display similar sensitivity to temperature as radiotherapy does, with a better effect for higher temperatures. And we validated that also for drugs used in high-tech procedures. So some of these effects are synergistic on a molecular basis, some are purely additive, so the optimal schedule also depends on that synergy. So the rationale is very strong for the combination. The outcome of the treatment does depend on the timing sequence, but mainly on the temperature achieved. And this synergistic effect is already achieved at very moderate temperatures, which can easily be achieved also in the clinical setting. And that summarizes also my presentation to really convince you of the benefits of combining these two treatments. It's currently widely used, and we are now also looking at the next step of combining with immune therapy. I thank you for your attention.
Thank you, Professor Sands. If there are any one or two questions, then we can take it now. Professor Sands, I will start one question. This hypothermia in a low LET radiation is quite synergistic, and it helps, in forming the double strand break clusters, and it is quite effective. But if it's the high LET radiation, will hyperthermia be useful or not?
I think that if you look at more treatment-resistant tumors, it might actually still display some benefit, but way less than combining with low lead radiation. I think the main benefit of combining hypothermia with radiation is in the low lead radiation. But for instance, combining with proton therapy would still give benefits because there the type of damage induced would still be enhanced by combining with hypothermia.
Proton is relatively low LET.
Yes,
only,
yeah.
But what about the carbon?
I think for carbon it would not be very logical to combine. No.
Okay. Any other question? Yeah, there's one there. So you want?
Thank you, Hans, for a very nice talk. And I'm very curious about the sequence of the therapy. So you show that hyperthermia therapy, and then just after radiation therapy is a very effective way. And then the duration between this sequence should
Yeah.
be very,
Yeah.
very close, yeah? But
Yeah.
not so short, yeah?
Not there are what we normally do in the clinic is we aim for a time interval between 30 and 60 minutes, and we see no negative effects in the normal tissue. There is one group in Switzerland where they are advocating first giving hypothermia and then within a few minutes give the radiotherapy, and they do that with a lower radiotherapy dose. So they give, I think, 5 times 4 gray, which is 20 in total, and they achieve very good results without any toxicity to the normal tissue. So we are also applying that, but I have to add there that they implemented that particular schedule without too much prior basic research. So, and I think it's a topic that also should see more attention because there is certainly a lot of benefit for applying that.
Thank you. Dr. Hans, I know what Hans here. What we practice is when the patient is waiting and for the radiation to take, either prior, that means when the other persons are waiting, and we start the hypothermia for this patient, and by that time another 15 to 20 minutes later, and this patient will be taken, and it will be weekly, 3 times a week, and it's being practiced, and for about 2 to 3 weeks in a period. That's the way that is being given, not on the first week, but on the second week later, second, third, and fourth week of radiation. Is that okay?
I think that's okay, yes.
Okay, fine, thanks. Thank you. Yeah.
Thanks, Dr. Hans. So now our second speaker is Dr. Ashish Ranjan. He is from UT's Southwest Medical Center, USA, and his topics is HIFU, West Thermal Ablation and Histotripsy, potential for solid tumor therapy. Dr. Ashish, please start it.
Thank you very much for the kind introduction, and thanks to the organizer for inviting me here. Actually, Bihar and Jharkhand happens to be my home state, so it's a homecoming for me in many ways. What I'll be doing today is a little bit of a lateral approach to treating solid tumors, which is not using radiation, but using sound waves which we call it as high intensity focused ultrasound or HIFU. And I'll be giving you some perspectives of how we do so in a bench to clinic setting using veterinary patients. The research that I'll be presenting today has been supported by several agencies. Those include the intramural and extramural program from the National Institute of Health, industry partners, as well as foundations who support our research in many ways. So as I was telling to you all that the kind of solid tumor treatments that we do is using a lateral or a distinctly different approach than how you might treat tumors with radiation. And we do so using sound waves, which is produced by an ultrasound transducer. And if you think of ultrasound transducers in general, sound waves are emitted from the transducer in parallel. But what we do with HIFU or high intensity focused ultrasound is we focus those sound waves or sound energies to a point in a tissue. And when we do so, we generate high mechanical and thermal energy, which can be leveraged to treat the solid tumors. This kind of treatment is done under image guidance, so you know where the sound waves are being transmitted and where they're hitting the tissue, and you can guide those treatments using ultrasound, using CT scan, using MRI, and you can even measure the mechanical or thermal effects in real time while the treatments are happening. And this idea of using sound waves, sound energy, using this HIFU modality has been really picking pace. More than 34 indications throughout the world has been approved for treatment with HIFU, and 7 in US have received FDA approvals for treatment using HIFU therapies. And when we think of treating a tumor with HIFU, we can use it to generate mild hyperthermia, which is heating tissues to 40 to 45 degrees Celsius in a non-cytotoxic manner, or we can even thermally ablate tumors, which would be heating tumors to a temperature greater than 60 degrees Celsius for a minute, or we can even mechanically treat solid tumors non-thermally. So you don't have to generate heat, but you generate high acoustic pressure in the tumor, and by doing so you can cause the tumor cells to lyse. And we call this technique as histotripsy, which is basically tissue lysis, tumor lysis, using this HIFU approach. So if a clinician wants to treat a solid tumor and he or she has a tumor that they would like to target, essentially the first step of HIFU treatment is locating that lesion. So in this particular case, I'm showing you a lesion here, which a clinician would want to treat. And then they'll select a dose. It could be a thermal dose, it could be histotripsy, it could be thermal ablation, and then they will combine the image and the treatment in real time using softwares, and that's how treatments are delivered locally in a very targeted manner. And that's why we also call this kind of treatment as image-guided therapy. You do treatments under image guidance in a very targeted manner. So HIFU can be used in many ways. As I told you, it can be used to generate cytotoxic heat. It can be used to generate mild hyperthermia. But one of the very novel findings that have been discovered in the recent years is HIFU's ability to activate the immune system against the cancer cells. We call this whole approach as induction of anti-tumor immunity using HIFU. And to test this idea, what we did was we started with mice model initially. We grew tumors in the flank region of the mice, and we grew bilateral tumors. So in the sense that mice had tumors on both sides of the flank. And then we treated one of the two tumors with HIFU. And the reason we did so is we wanted to understand if you just treat one of the two tumors with HIFU, does the untreated tumor go away? And if they go away, it would indicate an activation of the immune system because they are not being treated. So what we did was we compared 3 kinds of parameters. Those included hyperthermia, ablation, and histotripsy. And I won't go into the specifics of the parameters. This is a murine HIFU treatment system. There's a transducer that can be used to align with the mice tumor and can be used to treat, and you can see the treatments in real time in a computer. So when we treated mice using this approach, one of the first endpoint that we looked for was tumor remission. And what we noted was relative to control, the treated tumor regressed with the mechanical ablation treatment being more effective than thermal ablation or mild hyperthermia. But interestingly, all the untreated tumors, regardless of the source, whether they were histotrypsy, whether they were ablation, whether they were hyperthermia, regressed. So what it indicates is, while you can get cytotoxic effect against the treated tumor by treating tumors and re-engineering their immune environment, you can also generate a systemic anti-tumor effect that is evidenced by the remission of untreated tumors in those mice. And we went a step further and characterized the gene expression patterns in those tumors that are immune related. And you can do so using RNA-seq, immune specific panels that we use in this particular case, which we call it as a nanostring panel that measures 800 genes in one time that are immune related. And what we noted was each kind of treatment generated the immune outcomes in very specific way. When we looked at mild hyperthermia, it generated innate immune system and activated cells that would do antigen presentation for T cell activation. When we looked at the ablated tumor, they actually induce more pro-tumoral cytokines that would in fact cause enhanced tumor growth, but at the same time they also enhance the receptor activity of various immune cells in the tumor that might counteract the pro-tumoral activities of thermal ablation. And when we did histotrypsy, we noted that cells of cytotoxic T cell phenotypes got activated. These are the cells that are really good for anti-tumor immunity, and that's what we saw upregulated with this kind of treatment. And these were also reflected at the cell level, but what we noted was comparing these three modalities head to head, histotrypsy appeared to be more effective in inducing anti-tumor immunity relative to the other modalities that were being used. Now this is very exciting, but one challenge that we run into being rodent researchers at times is when you want to take rodent ideas into a human or to a veterinary patient, it becomes very challenging to translate. In fact, in US, you're looking at a 20 to 30 year timeline to take research from rodents into humans. So it's a challenge, and we need to have bridge models that can bridge things in a manner that speeds up the pace of clinical translation. And especially in the 21st century, there is also a lot of next generation therapeutics. You have nanomedicines, you have immunotherapies that is asking us to look beyond rodent models and choose models that are more translatable and that are more similar to what we can do in humans. And because of that idea, at the UT Southwestern Department of Radiation Oncology, we have now established a new division that we call it as Veterinary Research and Oncology Clinic that I lead. The goal of which is to enroll dogs and cats with cancer within the human hospital and do parallel trials in humans and in veterinary patients using this unique idea of one medicine. One medicine essentially is integrating veterinary and human medicine under one umbrella so that you can enhance the pace of translation of therapeutics faster. And if you think of canine patients, dogs, they get cancer. In fact, 1 in 4 dogs would get cancer in their lifetime. And in US, almost every home has 1 or 2 dogs. So you can think of the number of pets that get cancer and require treatment. But the very unique aspect of canine patients is the annual incidence, the site situations, and the tumor types are very similar to humans. In fact, there are some tumors in dogs that are exactly similar to what you may see in humans. For instance, I'm showing you dogs with oral melanoma here. It resembles dermal melanoma. So if you are doing trials in dogs with oral melanoma, you can be very confident of taking those therapies to humans faster. dogs with sarcoma resembles pediatric sarcoma in their totality. So again, you have a chance there to take therapies faster. So what we did, we started enrolling pet dogs and cats with cancer to our trials. And here I'm showing you a dog with an oral plasmacytoma that was going into the lower left lip, all up to the gums. And this tumor was a recurrent tumor. So the tumor was removed surgically and it came back. And here is how the tumor looked like in the ultrasound image. And we exposed them to HIFU, covering about 60% of the total tumor volume. And in 3 weeks, the tumor was gone and there was no evidence of any ulceration. And when we did the biopsy of those tumors, we could see infiltrations of immune cells that would be indicative of an activated immune microenvironment. This is another case of amyloblastoma, which is an odontogenic tumor that was growing into the lower left mandible, big tumor. Again, we treated this with 2 thermal ablation sessions, and the tumor was gone in 3 to 4 weeks. This is a particularly interesting case. This is a tumor that is a schwannoma. It's very similar to humans' nerve sheath tumor that is seen in patients. This was growing into the left upper maxilla, very big tumor, pretty hard to treat. And we enrolled this patient and exposed the tumors to a thermal ablative regimen for 3 times. And by week 7, there was no evidence of tumor present, and this dog remained tumor-free for 2 years. All of these indicate that when it comes to HIFU, it has potentials for treating oral tumors in humans, and if we can leverage some of the knowledge that we're gathering from veterinary patients, those could be very crucial. And those are also reflected in their mechanistic patterns with treated tumors showing significant inflammation, T-cell inflammation, both in the blood and biopsy over time. So one question that we did not know is, are all ablation treatments same, or are there any differences between the type of modality? And also whether the immune effects are tumor agnostic, because it's possible in some tumor types you show more immunity, in others you don't. So what we did was we did a very small pilot trial where we recruited 5 patients each for thermal ablation and histotripsy exposures independent of their tumor type. And we also compared them with mild hyperthermia in combination with dogs. This slide is showing you the parameters that were used to treat the patients. And what we noted was when we treated the dogs that had tumors of different types, mast cell tumor, mammary mass, apocrine gland, carcinoma, fibroma, in majority of the cases, even when we treat 60% of the total tumor volume with thermal ablation, we could see a complete response up until 60 days of monitoring, which was quite interesting. However, those kinds of treatments were also associated with some adverse effects, skin burns, GI symptoms. So while the treatments were effective, there was some adverse effect which should be expected because you're using a thermal ablative regimen. And when we looked at the tumor, we could see significant coagulative necrosis, which was indicating that the treatment was very cytotoxic mechanistically, but it was delivering the outcome in the manner you would expect. However, when we did histotripsy, we covered about 30 to 50% of the total volume. What we noted was only a few dogs showed partial response with majority progressing over time, which would indicate that unlike thermal ablation, histotripsy by itself as monotherapy may not be used as a sole therapy. But regardless, when we look at the immune features in the blood, especially the immune cells that would be considered functionally active, that would be considered active against a tumor cell, we noted that both the CD4 cells and the CD8 cells that are interferon gamma positive, where the population of them were enhanced in the blood samples. And also when we looked at the cytokine profiles in the serum, we noted that cytokines that would indicate an enhanced tumor progression in an immune-mediated manner were reduced. For instance, GM-CSF, IL-7, IL-18, all of them are pro-tumoral cytokines, and both were reduced in the responders, whereas cytokines such as MCP1 and IL-8 that would indicate a progression in an immune-mediated manner in non-responders increased with these treatments, suggesting that these treatments are linked, the immunomodulatory effects of these treatments are linked with the HIFU parameters. Now, as we found from our murine data that mild hypothermia by itself may not be effective, so we combined mild hyperthermia with doxorubicin, and we chose 3 sarcoma patients as our enrollment subjects. And we did sarcoma patients because sarcoma patients are sensitive to doxorubicin, so we thought, why not see whether there's any synergism present. And what we did, we chose 2 regions of the tumor, one region being heated, the other region being not heated. And obviously the heated regions had more drug due to the presence of heat. We didn't follow the patients for a long time, but we saw response in 2 of the patients post-treatment. But interestingly, when we combine mild hyperthermia with doxorubicin, we see a significant uptick in the population of CD3 plus cells in the tumor masses, again suggesting that combining heat that is HIFU based with chemotherapy could have synergistic benefits for patients. So in summary, what I showed to you in my last 15 to 17 minutes of talk is HIFU can achieve tumor growth control like radiation, and especially some kinds of HIFU like histotripsy induce no adverse effect in the patients. It's fairly easy to use it. You can do that in an image-guided manner. But more importantly, HIFU has an immunogenic effect, and that could be very critical if you're thinking of doing immunotherapy trials, or even if you're thinking of combining HIFU with chemotherapies to induce an anti-tumor immune response in the patient. Right now, we have a trial going on in larger cohorts of patients at our medical center, and hopefully if I get to come back again next year, I'll have the opportunity to present that. Thank you very much.
Thanks, Dr. Ashish Ranjan. And very nicely covered. Any questions from the floor? Yeah.
Great talk. Yeah, I thoroughly enjoyed your comparative oncology approach, which is very vital because oftentimes we forget the objective is not to treat mice. The objective is to treat humans. So that's where your dog model is the way to go.
Yeah, thank you.
Now, question, when you do HIFU post-treatment, have you tried to measure other cytokines, particularly to see if there is an elevation of anti-tumor cytokines such as IL-12? And have you tried to see whether there is a change in the macrophage phenotype from pro-tumor M2 macrophage to the anti-tumor M1 macrophage? These are important questions because most, if not all, radiation therapies lead to the elevation of NF-kappa B, which is a pro-tumor signaling pathway. Likewise, most radiation therapies lead to enhancement of pro-tumor IL-10 cytokine. That's why those patients undergoing radiation therapy often need to be treated with immune boosting drugs actually. So just I want to see if HIFU has a potential to reduce the production of pro-tumor cytokines.
That's a great question. So what we do when it comes to doc trials, we use a biplex array. It's a collection of 23 cytokines, and the data that I showed to you are the cytokines that were detectable. So one challenge that you run into the dog world is the immunological reagents that are widely available for humans and rodents are not as widely available for dogs. But we did a 23 cytokine panel, and the ones that I showed you were the ones that were showing sensitivity in the assay. Now, when we do murine trials, we always see M1 phenotype in the tumor. So that's one of the major boosting effect of HIFU. It can reverse the M2 to M1 phenotype, and not just that, it can also change the MDSCs, not only in the tumor, even in the liver. So there's a connection there. But unfortunately, in the dog world, we still don't have antibodies available to characterize those cell phenotypes. That's why we have been relying mostly on the T cell phenotype and nanostring to complement that. So we are working on a panel. I can share with you. Like right now, we have a panel that is about 16 colors. So hopefully, we'll have some of that optimized in the months, if not years, to come.
Any more questions?
Sure. The 2 points. One is, is it possible to categorize the tumor that you take for the experiment, like which are tumors which are immunogenic, and measuring a simple like PDL1, MSI, and TDL ratio, to see whether it's a tumor, it's a hot or cold tumor, and those things will easily help you to go ahead and with this experiment one. And another one is, irrespective of the type of the insult that we do to the tumor cells, ultimately what we are really releasing is a tumor necrosis factor that gets released. Whether it's a radiation, high dose radiation, or whether it's HIFU that you want to do. So therefore the pathway is the same at the end. So why do you want to again reduplicate the same thing and say that from animal model we'll have to take it to the human? Is it really necessary? Because radiation is seen. It is practice actually, right? Right now it's in the clinical use. Go ahead.
Yeah, so those are two great advices. The suggestion Dr. Vilimoga was making is, can you categorize tumors into poorly immunogenic, which are cold tumors that are not inflamed, that don't have immune cells, and hot tumors that are already inflamed, and would the responses be any different between cold and hot tumors? So all of our murine work actually focuses on poorly immunogenic tumors because hot tumors you can treat with any modality you feel like, but when you think of cold tumors, that's where you need to re-engineer. And even in the dog world, you can categorize hot and cold tumors. For instance, melanoma sarcoma would be considered cold tumors. And the reason we pick cold tumors over hot tumors is one of our key interests is to combine them with anti-PD-1, anti-PD-L1, or anti-CTLA-4. So all of our studies are focused on that, using that. Now, HIFU is not just a necrosis releasing approach. It also induces a type of cell death that we call it as immunogenic cell death. And that kind of cell death is characterized by upregulation of calreticulin, HMGB1, ATP release. So by tailoring the HIFU parameters, you can get that kind of cell death that is immune active. So that's why we are focusing on some of those to make them not just necrosis dependent, but also immunogenic cell death dependent.
Any more questions? If no questions, then thanks to Dr. Ashish.
Thank you.
It's my proud privilege to invite our next speaker. She is a well-known giant figure in the field of Indian radiation oncology. She is Professor and Head, Department of Radiation Oncology, Aiims Patna, and the organizing secretary of this event, Professor Dr. Pritanjali Singh ma'am, for her talk on low-dose radiation in COVID-19 pneumonia, a resurgent science.
Thank you very much, Chairpersons. At the outset, I would like to welcome you all to this very important international conference on radiation research, the impact on human health and environment, and the 4th biennial meeting of the Society of Radiation Research. As the Organizing Secretary at Patna. Welcome again to the land of enlightenment, Patliputra. So I will take you through the course of our journey during the COVID times. As this small topic, I will be discussing low-dose radiation in COVID pneumonia, which is indeed a resurgent science. So when COVID-19 was declared a pandemic in 2020, March 11th, it followed unprecedented global death and disruption, and we were all witness to it. Most of the patients that had an indolent course were the mild ones, but advanced age as well as comorbidities led a higher risk of respiratory failure. And the mortality rate was as high as 30 to 50, 80% once the patients were dependent on mechanical ventilation. And we all were, of course, a witness again to this. The SARS-CoV-2 viral particles infected the alveolar type 2 pneumonocytes, and they produced a cascading inflammatory event mediated by a cascading hyperinflammatory macrophage activation event in the lungs. As you can see in the alveoli over here, you can see that the edema was being formed. This cytokine storm led to pulmonary edema, infiltrative inflammatory cells, diffuse alveolar damage, and injury to the myocardium and extrapulmonary organs as well. It was indeed very devastating. So we were going through many articles, and this very article by Edward Calabrese on how radiotherapy was historically used to treat pneumonia, could it be useful today, really hit our heads. In this article, there were many references to radiotherapy that had broadly been accepted by the Radiological Committee for starting in the second decade of 20th century, with notable successes in the treatment of a wide range of inflammatory and infectious diseases as well, such as gas gangrene, carbuncles, sinusitis, arthritis, and inner ear infections. The initial report of using the X-rays to treat patients with pneumonia was far back in 1905 by Musa and Edsall at the University of Pennsylvania. And Quimby also had reported on the successful treatments of 12 cases of unresolved pneumonia. In 1924, Haydenhan and Fred simulated research on the effects of X-rays on the clinical course of pneumonia, and they reported on 243 acute and subacute pyrogenic infections of numerous types which were treated with X-rays. This paper offered a new mechanistic insight in the dose response, proposing that low-dose radiation would be an effective in treatment of pneumonia and disease resolution. The X-ray therapy was used to treat pneumonia, of course, in many patients, and we found that there were more than 15 studies that reported approximately more than 700 cases of bacterial pneumonia, be it lobar or bronchopneumonia, which were sulfonamide non-responsive, interstitial and atypical types, and they were effectively treated by low-dose X-rays, leading to disease resolution based on clinical symptoms, objective disease biomarkers, and mortality incidence. The capacity of the X-ray treatment to reduce mortality was similar to that of serum therapy and sulfonamide therapy during the same time period. But as everyone would know it, thereafter came the era of antibiotics, and this science was died down slowly. But we had reports of so many cases being treated by various authors, and they were, as you can see in the chart, case numbers and the cases cured. The cases cured were numerous. So this led us to sit and talk and discuss what exactly led, and this review also said that actually radiation led to an anti-inflammatory effect of low-dose radiation therapy because, first, it resulted in apoptosis and decreased adhesion of the leukocytes to the endothelial cells, which was the first step in causing the inflammatory process. Secondly, it mitigated the pro-inflammatory effects of the macrophages, reducing the secretion of nitric oxide and reactive oxygen species. Then, it also led to polarization of the M1 inflammatory macrophages to the M2 anti-inflammatory subtypes. Furthermore, LDRT also reduced the tumor necrotic factor alpha, interleukin 6, interleukin 2, interleukin 16, interleukin 8, and interferon gamma. And this all were thought to be increased in COVID-19 cytokine storm. So if we could address the cytokine storm at that time, then we could lead to resolution of the whole thing. Hence, it was hypothesized that low-dose radiation directed to the lungs may dampen the cytokine hyperactivation and improve the outcomes of the COVID-19 hospitalized and oxygen-dependent patients. Ours hospital was a dedicated COVID hospital from June 2020 to January 21, in which we were only treating COVID patients. The whole wards were filled with COVID patients. Be an oncologist or be a surgeon, everybody was into the treatment of COVID. And hence, we sat down in our department and we introduced this small discussion or a study of the impact of adding low-dose pulmonary radiotherapy to moderate COVID-19 pneumonia patients. This is the IMPACT RT study. Ours was the Department of Radiation Oncology, but we had collaborators from the Department of Pulmonary Medicine, from Radiology, from then Medicine as well, and of course anesthesiology. So we all combinedly sat down for the protocol writing, which was in a very finer way, being it feasible, being it interesting, novel technique, ethical was very much important and relevant. And then we went over literatures of various things, then found the evidence, we jotted down our endpoints, the research questions that we would want to formulate, protocol writing then followed, and of course peer review, and then the final version came out. We then went through the DRC, that is the Departmental Research Committee, and the Institute Research Committee, then the Ethics Committee, and finally we had institute approval, and we had also intramural funding, which helped us to take up this small study of ours in our patients. So again, I would like to emphasize that LDRT to the lungs led to polarization of macrophages towards the anti-inflammatory M2 phenotype, which helped in decreasing the inflammatory process. And it is with this thought in mind that we thought that we would give it to our patients of moderate pneumonia defined by the ministry. So the LDRT can also could prevent the cytokine storms by lymphocyte exhaustion and immune consumption. So with this, of course, this prospective randomized clinical trial was taken up, and we studied the role of LDRT on the moderately severe COVID-19 pneumonia patients with the evaluation of clinical, biochemical, as well as radiological parameters. The objective of the study was primary objective was to evaluate the changes of the moderate to severe grade of disease, that is increasing the severity of the disease in both the groups, the two randomized groups with NEWS2, which is a physiological parameter in time frame of day 3, day 7, and day 14. And the secondary objective was to evaluate the changes in the physiological parameters, including the oxygen levels, SpO2 and the FiO2, the laboratory testing with WBC, platelets, ESR, D-dimers, LDH, ferritin, interleukin-6 and CRP levels. The CT thorax was also performed on the day 1 and day 7 of the study. Radiation-induced acute and late toxicity was thought to be monitored on the basis of the CTCA-5 scale. And we wanted to see how many patients converted from non-invasive into intubation after following the LDRT course. The inclusion criteria included patients more than 18 years of age to 70 years of age, patients with confirmed diagnosis of SARS-CoV-2 infection by RT-PCR on the nasopharyngeal swab and on bronchoalveolar lavage, patients who had Ministry of Health and Family Welfare moderate disease with SpO2 level of less than 94 at room air and respiratory rate of more than 20 breaths per minute on room air with fewer than 10 days of symptoms. This was important because this was when we could resolve the disease, and if it was too long, then maybe the radiation would not work. So suggestive picture finding for interstitial pneumonia on the chest X-ray and chest CT was optional, and ability to understand and sign the informed consent was of pivotal importance. Ability to acquire and maintain the setup necessary for the delivery of radiotherapy was also very important. Exclusion was of those patients who had severe disease, who had invasive mechanical ventilation, who were active autoimmune systemic diseases, who were hemodynamically unstable patients with low blood pressure, and patients with a positive pregnancy test, and those patients who are impossible to maintain the setup necessary for the radiotherapy treatment. So the study was, as I said, prospective, monocentric, double-armed, interventional, randomized, non-pharmacological, exploratory study 1:1 randomization. Sample size we thought we would take is 22. The project implementation plan in our study was multidisciplinary team, and we would all select the patients very subjectively according to the inclusion and the exclusion criteria. And the study was continued from May 21, August till 20, to August 21 at the All India Institute of Medical Sciences, Patna. So data analysis was done by the SPSS, clinical characteristic and demographic data was defined, and of course the mean and the median was there for the categorically variables, whereas the continuous and the other data were compared with the two tailored Fisher extract tests. So this was the flow chart. As you can see, first counseling was done, followed by consent, followed by randomization. The patients were treated as per the institutional COVID-19 treatment guidelines in both the arms together, but in arm 1, they received low-dose radiation, and in arm B, they did not receive any radiation. So this was the flow chart, and RT would be delivered to bilateral lungs on the same day after CT simulation. Dose was supposed to be 0.7 gray. Clinical and laboratory parameters were to be taken out at day 1, 3, 7, 14, 28, and 6 months from the treatment. Radiation would be considered day 1, and CT of the thorax would be done on day 1 and day 7. So this is the NEWS 2 score, which is a physiological parameter which tells us about the sickness of the patient, and we defined all the NEWS, basically all the patients according to the NEWS 2 score on all the days, that is, which depends on the respiratory rate, the SpO2 level, on air or oxygen, the systolic blood pressure, the pulse rate of the patient, the consciousness level of the patient, and the temperature of the patient. So workflow was very important because it was difficult to transport the patient to the radiation unit. Immobilization and CT scan was done for all our patients, as well as contouring and treatment planning, and delivery of treatment was done with monitoring, and transport of the patient was then had to be done back to the ward. As you can see, the corridors were all clear, nobody would be there except for the people that were transporting the patient and the patient itself. We delivered low-dose radiation to bilateral lungs, as you can see, with the 3D CRT technique, and you can see over here that the patient is lying in the machine area and is constantly being monitored by the monitor which is inside with the patient, and single exposure with photons to bilateral lungs was delivered with an energy of 6 to 10 MV, dose 0.7 gray only at SAD and 3D RT techniques. So transport of the patient, as I said, very difficult because the whole corridor had to be evacuated, and anyways during COVID times there were less of people everywhere, and continuous monitoring had to be done by our junior residents and the senior residents that were there. And of course the patient had to be transported back to the wards as well. So this was a consort diagram. We had evaluated 68 patients, out of which excluded 55, and because 50 of them declined to participate, and others reasons were about 5. Randomized patients about 13, out of which patients allocated to the group 1 was 7, and patients allocated to group 2 was only 6. And they went low dose RT and follow-up as defined. And you can see that the distribution of the patients in both the arms were equal, and the number of days in the hospital and the oxygen was also equal. So what we saw is that on the 3rd day of radiation, the FI2 levels and the SpO2 levels did fall, and the levels were under showed statistical significance, but that was not maintained on day 7 and day 15 thereafter. Again, the same charts showing that change of NEWS 2 score in the RT and the non-RT arm on day 1 and serial follow-up over here. And this was not, however, statistical significance, but there was a change in the mean of FiO2 in the RT and non-RT arm just on day 3, as you can see from the statistical significance level over here. Remdesivir was given to 2 patients in the RT arm and 4 patients in the non-RT arm for whatever effect it would have on the disease. A decrease in the level of the biochemical markers of acute phase reactants was seen in the RT arm and the non-RT arm together. However, it failed to achieve the clinical significance on comparison between the 2 arms. There was no statistical difference in the decrease in the interleukin-6 between the 2 groups. The ratio was decreased on the 3rd day of the LDRT arm, which is similar to other prospective studies, but on days 7 and 14 of the hospital stay, this significance was lost. The median duration of hospital stay was 14 days for the RT arm and also for the non-RT arm. So although there was a trend towards early improvement, the difference did not show any statistical difference between the two arms after the third day. There was no lymphopenia and no added deterioration in the LDRT arm. There was radiation-induced acute pulmonary toxicity was not observed, and one patient in the RT arm requiring mechanical ventilation after clinical recovery developed sinonasal mucormycosis, and the patient died in the ICU, whereas a sudden cardiac death was also reported in the non-RT arm. And this picture, you can see that day 1 of radiation and day 7 of radiation, there's a marked difference in these two patients, subsequent patients that I am showing. And we had these on the CT scans, but this still was not statistically significant. So to discuss, there are many clinical studies that have evaluated the role of low-dose RT to lungs in the COVID-19 pneumonia. Most of them are single-arm studies and have documented feasibility, tolerability, and clinical benefit. However, very few studies such as that of Papa Christopher et al. Demonstrated no benefit of low-dose RT to improve the recovery in critically ill patients, severe COVID pneumonia requiring mechanical ventilation for COVID-19 pneumonia. So we had proposed this in moderate pneumonia so that it could not be converted into the severe type. The dose was given 0.7 gray, and according to recent evaluation, the lifetime attributable risk of cancer for a dose of 0.5 gray for the ages 20 to 80 can range from 0.29 to 1.9 for men and 0.5 to 4.9% for women. Cardiac events also after 5 years of radiotherapy, such as in the study by Darby et al., have shown linear relationship with increasing dose by 7.4 gray percent per gray, and Taylor et al. Have also shown that after 10 years of radiation, 0.04% per gray was increasing the level of mortality with cardiac events. So we had to, of course, follow up these patients and monitor our patients regularly. And these are the studies that I have enumerated. Most of them showed that there was an increase in everything, decrease in the improvement in the SpO2 levels, but the limitations was that they were small sample size studies. As you can see, these studies, many studies were going on. Ours was a double randomized, double arm study. There was improvement in the NEWS2 score, but of course, again, we were only able to accrue 13 patients. We are following up these patients at 6 monthly intervals. The history of addictions like smoking and background comorbidities, etc., enable the chance of attributable cancer and coronary heart disease to be increased. CT-based planning was done, of course, and the adjacent areas of radiation, whatever dose was delivered, could be chalked out for these patients. So this is a unique opportunity to measure such dosimetric data, which will serve as an important archive on longer follow-up if any recipient of low-dose RT ever develops the possible attributable side effects in lifetime. Right now, all the 6 patients that were radiated are good. They are doing well. Sample size was small. These were the limitations. And of course, the CT score was, the radiologist that was doing the CT scoring was blinded, but the treating physicians like us, we were not blinded. Then the course of COVID-19 pneumonia might improve itself. So we don't know exactly how much of role the low-dose radiation had played. Similarly, patients with COVID pneumonia respond differently to the standard protocol of treatment, and we were giving the standard protocol of treatment in both the arms. Hence, it is expected that LDRT will not produce a similar response to all patients in the cohort, although we meticulously tried to include moderately affected patients alone. We had challenges, as you can see. Ethical approval was difficult. Right patient recruitment was very, very difficult. Delivering LDRT at the appropriate time, informed consent from the patients, limited manpower, sterilizing the CT simulation and the LINAC machines after treatment, minimizing the contact of the healthcare personnel and other patients, and review of the patients, especially in lockdown period, was very difficult. And as you can see, the glaring of the lights in the CT simulation room, because the mobile covers were also covered with plastic. So it was very difficult. But to conclude, I would like to say that LDRT seems to have the potential to prevent moderate COVID-19 pneumonia from deteriorating to severe category. However, further randomized clinical trials with greater number of such patients is warranted to establish the definitive role of low-dose RT in the management of COVID-19 pneumonia. These are my references, and I would like to sincerely thank the group that was there with us, little manpower that we had, our residents as well as our acad-JRs. Thank you very much for your hearing.
saying purposely, anybody who worked during the COVID time, give me sir. Anybody who worked during the COVID time, whatever may be the small help which is towards the patient is remarkable. Our hands off to you. Okay, that is the one, the way that we really, appreciate your work.
Thank you so much.
Sir, you can come, sir. Sir, you can take it. You can take it. Misha Saab, yeah. Misha has some questions.
Yeah, Dr. Pritanjali, I must congratulate you
Thank you so
for
much.
making this initiative, because I recall many articles came in many journals suggesting that low-dose radiation could be a remedy for this kind of COVID infections, and I was one who was supporting it. I also wrote an article in our journal, Journal of Cancer and Radiation Research, and I pleaded that some medical people should have courage to initiate this research.
Sure, sir.
So I quite appreciate and I see there is a positive outcome, and in desperation,
It was desperation.
even though there is a risk, even though there is no knowledge that what may be the side effect, but you did this and commendable. I
Thank you so much,
appreciate.
sir. Thank you so much.
But the group at Bhabha Atomic Research Center, Dr. Pandey and Dr. Amit Kumar, would like to collaborate with you, because
Sure, sir.
we actively are looking into this aspect
Yes, sir.
of low-dose
Definitely,
radiation
sir. I would be in
application.
touch. I would
Yeah.
definitely be in
By
touch.
the way, I just also want to
Yes, sir.
inform you
Yes.
that Dr. Dinsha,
Yes, sir.
the then director of Tata Memorial
sir.
Hospital,
Yes, sir.
had approved one of the projects on human trial of low-dose therapy way back in 2005. And Dr. Pandey and Dr. Amit Kumar were to continue. Unfortunately, she fell ill and the whole program could not proceed further.
Okay.
But it has potential.
Yes.
I
Sure, sir.
deeply appreciate your effort.
Thank you so much, sir.
Thank you.
Thank you.
Okay, thank you, Python. Yes, sir. Yeah, come.
Yeah, I would like to echo the same sentiments. This is a clinical protocol
Yes, sir.
generally considered impossible in most parts of the world.
Sure.
And you and your colleagues
Yeah.
have made it happen. This is such a difficult clinical protocol. And we have been working serendipitously, because our area is not antibiotics, but we have been working on antibiotics as well as anti-COVID. One of our anti-COVID products was actually taken up by the Prime Minister's of India's office, Mr. Narendra Modi. I don't live here, I live in the US, so it's there all across India in the newspapers. But my question is, low-dose radiation, of course, it has been considered, as you beautifully showed us lots of literature. There is a possibility that it can work, mainly because the pathogen which is responsible for pneumonia, generally speaking, which is a Pseudomonas, it is not available on the surface. It builds a biofilm. Once it is underneath the biofilm, the antibiotics will not work, because antibiotics given through IV drip, they won't penetrate the biofilm. That's why we need an alternative approach, as you nicely described. Low-dose radiation might work.
Yes, we can see in that subset of patients as well, but of course ethical approval will be a big issue.
Yes, yeah. But the trouble with the low-dose radiation is that, as you showed it, it will enhance your M2, which is the
M2, yes.
Yeah, when you enhance M2 in the body,
Yeah.
humans become susceptible to
More infections.
secondary
Infections.
infections.
Yeah, sure.
So you have got to be very careful. It's a catch-22. Already the patient is suffering from COVID. There is the body is compromised in terms of the immune system. Then you're introducing insult to the injury by radiating the lungs. So that's something one has to keep in mind. Maybe the patient needs to be given, very workable, pro-immune boosting
So
drugs
if I can
alongside.
talk for my 6 patients, they didn't have any such sort of infection except for one that died of mucormycosis that I showed you. So if a larger subset would have been taken, maybe we could have come out with
Yeah,
something
you'll have to keep
statistical.
that in mind actually,
Yes,
because you're
yes,
making the
sure.
body susceptible
Sure.
for secondary infections. So some immune boosting agents alongside low dose radiation
Yeah,
might be a
all
way to
those
go.
were going on, vitamin D, vitamin C, and everything was going on as the institution protocol for COVID pneumonia.
Yeah.
All those were being taken
Yeah,
care
I
of.
think I see lots of synergies in terms of what we are doing both for COVID as well as pneumonia. I would like to
Yeah.
talk with you later on.
Okay.
Thank you.
Thank you so much.
Sir, this work was never taken as a standalone, sir. And it's an additive in addition to what
Yeah.
the protocol has already been given by the government of India. In fact, nobody has any right on changing that. It's a protocol which was followed.
Yes.
Every
Yes, the
every
protocol
place.
was being followed
In addition
in
to
all
that,
patients.
whatever the hypothesis says to improve it, and that's only added, and that was permitted, not other one. And in fact, even at that point of time, even taking doing the randomized trial was also curtailed to quite a large extent. There was no need to go through that. The hypothesis-based trial was permitted even
Yes.
at that point of time
Yes, sir.
because of the
Definitely.
emergency emergent period that
Yeah.
was in O.
So most of
Thank
the other,
you.
yeah,
Thank you,
sir.
Vaishnavi.
Thank you so
We
much.
will close this talk and we'll move on to
Thank
the
you.
next one. Next is from Dr. Gautam Sharma from Aiims Guwahati. Interesting talk. Actually, looks like it's more a continuation of what we know last time we heard, and one of that is on the exploring the lattice radiotherapy, clinical benefit and modern applications. And maybe now Dr. Ashish Ranjan, you can also very closely hear this, because in lattice radiotherapy and the partial HIFU in a particular organ has got a similarity in this. You can really closely observe this. Go ahead, sir.
Good morning, everyone. I thank the organizers for giving me this opportunity. So I will be talking on lattice radiotherapy, and my flow of the presentation will be like I will be talking on especially fractionated radiation therapy, which is lattice, and its evolution from 2D to microbeam, just a little bit of radiobiology and some clinical scenarios. So especially fractionated radiation therapy has a history of over 100 years, and this treatment of tumor happens with non-uniform dose. Actually, whenever we treat a tumor, we try to give a homogeneous dose. We doesn't want any hot spot or cold spot to be there in the treatment volume. And the dose range from 10 to 20 gray per fraction, and the application of SFRT historically known as grid therapy. So the original technique of grid therapy was developed in 1909 by Kohler, and initially it was used for skin cancers to increase the tolerance of the skin. And grid collimators deliberately creates hot and cold spots inside the target volume, that is called dose heterogeneity, and usually used for a single fraction. And in 1950s, SFRT was initially delivered via orthovoltage X-rays to treat deep-seated advanced bulky tumors. So you can see in the picture like a homogeneous beam is coming, and then a grid collimator is placed. And you can see that these grid collimators are not straight. They are designed in such a way so that it matches with the beam divergence. So 2D grid therapy was delivered using a conventional LINAC with a grid block. And to find out what is the most optimal hole diameter, so Monte Carlo simulation was done for 25 different grid patterns, and they found that 1 centimeter to 1.5 centimeter with spacing of 1 to 1.7 to 1.8 centimeter is optimal. So from 1990s, grid therapy was delivered using mega voltage photon beams to treat patients with massive or recurrent tumors. So after that, many clinical reports has shown good oncological outcomes using the grid therapy. So in the 3D era, the extension of this 2D grid therapy is known as 3D or your lattice radiotherapy. So it is the creation of multiple localized high-dose small spheres called vertices with a separation within the tumor volume, while keeping the dose level lower in the periphery of the tumor to avoid toxicity. And LRT is based on two things. Number one, that the proven clinical effectiveness of grid therapy that has already been published, and the new advances in physics and techniques that allowed creating 3D high-dose regions concentrated in small spheres called vertices inside the tumor volume possible. So now in 3D, MLCs are being used to create this grid, and this is a technique called IMRT. So IMRT has been used and MLC-based grid is developed. And this is a grid using the VMAT technique. So the basic difference between 2D and 3D grid is that in 2D grid, we cannot conform our dose to the tumor volume, whereas by using the lattice radiotherapy, we can conform our dose to the tumor volume. Now even grid therapy can be delivered by using tomotherapy also, and it has the advantage because here we can select the pattern and the shapes we need to treat certain type of tumor. So helical tomo grid can provide equivalent or better treatment plans as compared to LINAC grid, and it also allows 3D optimization to achieve superior sparing of the OAR and no high dose spill outside the grid GTV as compared to LINAC grid. So if you see the dotted line, this is what if we give a homogeneous dose to the tumor, and the peak and the valleys, it represents the dose profile of a grid therapy. Now coming to microbeam radiation therapy. Microbeam radiation therapy was invented by Slatkin in 1992 and is a form of SFRT using KV X-ray beams, the size of microns with widths of 25 to 50 microns and spacing around 200 to 400 microns. And the advantage is that this allows peak doses as high as 100 gray while sparing the majority of the normal tissue. Now, there are only two facilities in the world where microbeam radiation therapy is available, one in Australia and another one is France. Now, in the age of proton, so proton mini beam radiotherapy has come up. So it can be generated in two ways. Number one, by using a collimator with slatted parallel beamlets, and the slatted parallel beamlets are typically 300 to 1000 micrometer each with space of 1 to 4 millimeter apart on the center. And the second way, using the individual pencil beam dose painting methods to create a segmented beam profile. So we can see the difference between a conventional RT and a mini beam RT. So in conventional RT, most of the tissues will get the dose. So the area getting the, or the tissues getting the dose will be too much, and while this can be minimized by using lattice radiotherapy. So proton beam mini beam radiotherapy is now available in only 4 centers in the world, one in France, one in Germany, and 2 in US. So grid therapy is not a new thing, but it has evolved with time. So it started as 2D grid, and when the computers came, it became the lattice radiotherapy, and now we have our proton mini beam therapy. So the radiobiological mechanism, it involves multiple factors, including the vascular damage and the angiogenic response, radiation-induced immunoresponse, and the bystander and abscopal effect. So like why it is this importance has come up again after of such a old technique, because lattice radiotherapy uses 3D radiation therapy and IGRT and adaptive therapy to deliver high doses of radiation to small areas within the tumor called vertices. And LRT can limit the toxicity to the adjacent tissues, and LRT is useful for treating large tumors which are not typically treated with stereotactic body radiation therapy. Now I will go through some clinical reports. So this is the first case of inflammatory breast cancer. So inflammatory breast cancer is a very rare and aggressive type of tumor with very poor prognosis. And this lady was treated with lattice radiotherapy combined with fractionated external beam radiotherapy and concurrent trastuzumab. So what they found that at 1 year post-treatment, they found complete response. And it is the first documented case of inoperable inflammatory breast cancer successfully managed with lattice radiotherapy and systemic therapy. Then this is a paper for locally advanced CS cervix. So 10 patients were treated and the LRT dose was 24 gray in 3 fractions and it was followed by 44.28 gray with concurrent cisplatin. So the local control was 100% at median of 16 months follow-up and disease-specific survival was 53.3% at 2 years and the complete metabolic response was 88.9%. There were no more than grade 3 toxicity observed and this is a promising local control and tolerability was seen in by using LRT. However, the patient numbers were very low. Then this is a paper on head and neck cancer. So 14 patients of head and neck cancers with bulky tumors that is more than 6 centimeter were taken for study and grid radiotherapy that is lattice was delivered 20 gray in single fraction along with SIB IMRT. So what they found that the grid GTV control rate was of 79% and the acute skin toxicity of grade 3 and mucosal toxicity of grade 2 were seen. And the conclusion was that grid combined with SIB, IMRT and chemotherapy is tolerable and shows promising tumor control in bulky head and neck squamous cell carcinoma. So this is a paper where MRI guided LRT was used in 2 cases of liver metastasis and they found that the tumor response, interfractional tumor regression were observed. They didn't find any grade 3 or more toxicity and they said that the MR guided RT along with online adaptive LRT provides precise target localization and superior OR sparing, enabling the safe and effective delivery of high dose of radiotherapy to the voluminous liver metastasis. So this is a paper where LRT is used in the lung cancer. So they took 10 patients and the initial LRT fraction was 18 gray to the internal vertices and it was followed up with further conventional radiation of 25 to 29 fractions. So they found that average tumor volume reduction was 42%, median survival was 16 months and they didn't get any LRT related mortality. So efficacy wise LRT demonstrated promising tumor control in bulky non-small cell lung cancer and safety wise well tolerated with minimal side effects. Then in sarcomas 53 patients were treated using VMAT lattice and the patients got 20 gray and then it was followed up with consolidative EBRT of 40 gray. So they found the symptom relief in 60% of the treatment patients and stable or partial response in 90% and local control in 82% at 1 year. And the 1 year overall survival was 53%. So efficacy wise SFRT combined with EBRT demonstrated high local symptomatic improvement and favorable radiographic responses in sarcomas and safety wise the technique exhibits low toxicity profile with manageable side effects. Then lattice was added to immunotherapy to see the response. So this is a case report where high dose lattice therapy was combined with immunotherapy and they found complete response in that patient at 5 months and 77.84 regression was observed at 1 month. They didn't find any significant side effects. So they concluded that the case highlights the potential synergy between high dose lattice radiotherapy and immune checkpoint blockade for bulky tumors, demonstrating dramatic local control with no additional toxicity. Then lattice was compared between proton, carbonyl and photon and LRT with 1 centimeter spherical vertices were used and 15 gray of RB was delivered. So they didn't find any difference in peak to valley dose ratios. However, in organ protection, they found superior results in proton and carbon ion as compared with photon. So they concluded that proton and carbon ion LRT are feasible and offer better wire protection, making them promising options for sinonasal malignancies. Now this is a paper of metabolism guided lattice radiotherapy. So the vertices are placed based on the metabolic activity observed in PET scans, and 30 patients were taken in this study, and high dose radiation was delivered to the spherical deposits at areas with high metabolic activity, and the median dose was 15 gray in one fraction. So the results of these studies was that symptomatic response, 100% rate of symptom improvement, with 27 out of 30 patients showing response within 8 days. And immediate response was seen in 3 of 30 patients. And 89% overall clinical response rate, including 23% complete remission, was seen. Toxicity-wise, very few patients showed acute toxicities, and 86.4% 1-year overall survival was noted. They concluded that metabolism guided lattice radiotherapy is feasible, well tolerated technique offering impressive symptom relief and high clinical response in patients with stage 4 bulky disease. And these preliminary results suggest that this approach should become a leading palliative treatment option for patients with advanced bulky tumors. So, in conclusion, LRT has promising clinical outcomes, significant tumor shrinkage is seen, symptomatic relief, high clinical response, including complete remission in some cases. Safety profile-wise, very minimal toxicity with low incidence of severe side effects were seen in these studies. And MR-guided LRT improves tumor visualization and adaptive planning, and combination with immunotherapy enhances local control and overall outcomes in advanced tumors. So high potential for bulky tumors because it is very effective in treating bulky and radio-resistant tumors and providing palliative symptom relief. However, it requires further research to validate long-term efficacy and expand clinical applications. Thank you.
Paper is open for any questions. Okay, Gautam, it's a matter of more information to our audience regarding the lattice, even though it's a almost 70 years old technique, and how best we can even improve over that.
Yes, sir.
My only maybe comment is, it's good to have information. There's no doubt about that. We learn from the informations. But in Guwahati, you can get a number of patients like this in a university center. I really wish in next year you will come out with a paper and
Sure,
what
sir.
is your experience. That is where we are more interested in. Then it becomes the information applied to clinical work.
Yes,
Then we will really say yes, whether it's work or doesn't work in our setting. Okay, that's
Thank
my
you,
suggestion to you.
Thank you, sir.
Number 2 is, it is all applied in a bigger, large volume diseases. The major assumption is, for a large volume tumor, it's very, very difficult to give a very high dose of radiation, which we think that it's going to help in that setting. But if you give in a partial, either partial organ or a lattice one which can tolerate, and that is the way, and we can really give a required high dose, and immune system can be elicited out
Yes,
of the treatment area that we have done. In those 2 scores, how it has really come. It is not a curative treatment. There may be 1 or 2 instances that might have happened, but it's not a curative treatment at all. It is an adjuvant treatment along with the other modalities that we can really
Yes,
do.
sir.
Thank you very much.
Thank you, sir.
So in this series of hyperthermia, our next speaker is Dr. S.D. Sharma. He is at Bhava Atomic Research Center, Mumbai, and his topics is recent developments and update in flash radiotherapy. Dr. Sharma, please.
Thank you, chairpersons. At the outset, I would like to thank the organizing team for inviting me and giving me this opportunity. In fact, this flash therapy is a emerging technique. Whatever I am going to present, it is a literature review. We do not do any work as of now because we do not have any system available to us. So, as we know, radiotherapy is one of the important technique of cancer management. The goal of radiotherapy is always to deliver adequate dose to cancer cells and minimal dose to the surrounding normal tissues and organs at risk. The maximizing the tumor control probability and minimizing the normal tissue control probability is a very important role of radiotherapy. To fulfill these requirements, varieties of radiotherapy techniques and equipments are used, and a number of cancer cases are treated by these techniques. Even many developments has taken place in the recent past, starting from IMRT, BMAT, SBRT, and we are using the low dose radiation for treatment of these diseases. For example, we have available to us this spectrum of equipment which generates either gamma rays based radiation or high energy X-ray or electrons. So if we like to see what we have available to us today, we can categorize them in three categories. First, 2D technique based system like simple telecoupled machine. We use either a square field or rectangular field for treating majorly palliative cases. Then we have 3D techniques, that is 3D CRT, IMRT, image guided, volumetric modulated, adaptive radiotherapy, and then SRS, SRT, even SBRT, and even we have moved further to 4D techniques by incorporating the breathing pattern and monitoring the movement of the tumor with time. But if we like to say what is the major limitation of this technique, we are dealing with a conventional dose rate. Dose rate may be up to the value of about 5 to 6 gray per minute. So this is not thought to be a very sufficient dose rate for minimizing the dose to the normal tissues. So what we see, the overall limitation, this in case of present day radiotherapy, there is a lack of selectivity between tumor and normal tissue by the ionizing radiation, and this lack of selectivity inflicts acute and chronic toxicities to the normal tissue surrounding the tumor. Even radiation induces toxicities to normal tissue which limits the tumor dose, and this even limits our curative intent. The late toxicities resulting from the present day radiotherapy significantly impact the quality of life of long-term cancer survivors. So now these limitations are exciting us to look for something novel which eliminates all those limitations. So flash therapy is thought to be one of the potential candidate for fulfilling these requirements. So considering this, in 2014, Faudon and Bozinin coined the term flash irradiation and demonstrated that it possesses normal tissue sparing capability while maintaining the same tumor control probability or tumor cytotoxicity whatever we achieve by today radiotherapy techniques. The process includes delivery of dose to the tumor at a ultra high dose rate, not less than 40 gray per second, but practically it is about 100 gray per second, and it is much higher than in comparison to the dose rate we have with conventional radiotherapy techniques. This treatment approach induces the phenomena called flash effect, which reduces the normal tissue toxicities commonly associated with conventional radiotherapy technique while maintaining the same tumor control probability. This technique has the potential to revolutionize the future of cancer treatment, but there are some translational challenges, something I will bring to you. The ultra high dose rate radiotherapy or flash radiotherapy, what we call, is now very popularly known to the community which is utilizing this, but many systems are under fabrication. Prototypes has been brought forward for doing some trials, and many publications has been brought in the literature, but the excitement has been generated very recently because a few in vivo studies uh, mm, has been published very recently. So people are now gearing up to implement these techniques in the clinic with the resources whatever available to them. As far as the type of beams are concerned, electrons are also a potential candidate along with photon and high energy X-rays, whatever we are using today, but the energy shall be much higher than whatever we are having as of now. For example, if we see the curve, the depth dose curve for electron, we feel that electron has limited penetration power, but in case of photon, we see that the even build-up region dose and exit dose could be a problem. But if we look for the protons, probably we feel that build-up region dose could be a problem, but exit dose is not at all, and the very high dose can be given to a region of our interest. However, all these beams has been tried for exploring the flash radiotherapy with advent of some of the equipment. So, but before going much in detail, now we should understand how flash radiotherapy works. So, in case of accelerator-based radiotherapy sources, all the systems are pulse system. So, if we like to go in depth of the physics of this pulse system, you can see that pulse repetition frequency is usually of the order of 200, and the pulse duration is about 3 to 5 microsecond. So if we like to see the overall treatment dose, which is very, very less, but if we like to see what is the dose per pulse, that works out to be very, very high. So there are many parameters associated with analysis of the pulses generated by our accelerators, that is pulse width, the gap between pulses, number of pulses generated together, how many pulses will be generated in a second or in a minute. So if we combine together, earlier we were dealing with macro pulses, that is overall dose delivered to the tumor, but now in case of flash radiotherapy, we are focusing our attention towards the micro pulses. So this is another way of explaining the same thing. Here you can see that dose per pulse in our present day radiotherapy is of very, very less. However, in case of the flash radiotherapy system, the dose per pulse goes up to 100 gray per second. In another term, if you ask me the question, how we can enhance this capability of our existing accelerator beams. So if we increase the current of the beam, probably we may be able to achieve this goal. So in general, if we like to compare the conventional system available today and flash radiotherapy system, the pulse repetition frequency is about 300 pulses per second in our existing machine. However, we are looking for less number of pulses so that we can increase the dose rate. So finally, if you see, the dose rate in case of current day system goes up to the 56.7 gray per second only if we enhance the existing system. However, if we go for increasing the current, probably we can get the dose rate of the order of mega gray per seconds as well. So now this is another way of this radiobiologically what the process is. Here we usually say this radiobiological phenomena can be explained in three different factors: physical processes, physiochemical processes, and heterogeneous chemical processes. This is applicable both for flash radiotherapy as well as for conventional radiotherapy. So in this area, the knowledge is not very wide. People are still trying and they are collecting the information. So if we want to propose a hypothesis which is giving the birth of the flash effect, usually three hypotheses are acceptable as on today. It is called radiochemical depletion of oxygen, which is the currently most acceptable hypothesis. However, it has also been questioned by some researchers. Modified immune response, Then finally, the difference in the metabolic characteristics of the tumor and the normal cells. So first hypothesis, that is radiochemical depletion of oxygen. This is still a valid approach as of now. In this case, prominent role of oxygen is it generates reactive oxygen agent, species, which helps in creating this phenomena. And particularly, we know that if we analyze the microenvironment of the tumor, we see that the tumor is associated with the oxygen, a given level of the oxygen. But when we deliver the dose at a very high dose rate, it temporarily creates a hypoxia in the normal tissue. However, the tumor cells are itself having deficient of the oxygen concentration. Because of that, the normal cells develops transient radioresistance, which is helping them in getting spared by the radiation. However, the tumor cells are getting preferentially killed. So this is the hypothesis we are following as of now. However, it is the matter of elaborate research, and many people are working in this area. Schematically, if I want to present this case, if I need to present the radiosensitivity of the tumor cells and the normal tissue in with respect to the oxygen concentration or oxygen tension, we will see that once the oxygen tension is increasing, the radiosensitivity also increases. So the irradiation helps in minimizing the radiosensitivity of the normal cells. That is the basics, and it is the principle of the flash radiotherapy. So what is the current status of flash radiotherapy? Most of the studies has been used low energy electron beam, whatever was available with our current clinical accelerator. So it has limited penetration power. So people has started exploring the possibility of using proton beam, and some work has been done and published as well. But very many people has conducted theoretical studies, and they have proved that because the proton therapy systems could be a very costly equipment, so people are looking for very high energy electron beam. The energy range of these electron beams are of the order of 100 to 250 MeV, and they have found that the very high energy electron beams are also producing the similar effect whatever we are getting by the proton beam. So it is thought to be a light particle alternative of the heavy ion therapy for inducing the flash effect. Even very recently, the multiple direction X-ray beams has also been tried, and some works are going on in China, and they have come out with one accelerator which is called phaser, that is pluridirectional high energy agile scanning electronic radiotherapy system. This is thought to be a next generation of medical electron linear accelerator. However, if you see the design, it is very complex. Operating this accelerator will be a complex aspect once it comes to the practice. But the beauty is, it is a ultra rapid system. It is delivering dose by highly conformal way, and it is image guided system. It is fast enough to freeze the physiological motion of the tumor. It has the improved accuracy, precision, and potentially superior for delivering the flash radiotherapy, which helps in enhancing the therapeutic index. This system is, as of today, in comparison to the proton therapy system, thought to be a compact, clinically efficient, and even economical as well. So this is a schematic diagram of this system. It uses 16 medical linear accelerators with high dose rate options, which can induce the flash effect. The dose rate is about 400 times, the current is about 400 times larger in this accelerator in comparison to the existing clinical accelerator. The phaser platform is basically a X-ray system. It is fundamentally a new approach to deliver ultra-high, rapid, ultra-precise radiation treatment. But this system is not yet available in the practice. It is a theoretical simulation, and some reports has been published that some prototype fabrication work is in process. However, if we look from the clinical point of view and basic dosimetry point of view, this accelerator seems to be a system which fits to the purpose of the flash radiotherapy. Now, as far as the use of proton beam is concerned, a specific filter has been created for implementing this approach. This filter is called the patient-specific ridge filter for conformal flash proton therapy. This is a kind of personalized treatment delivered to the patient. However, this approach requires a lot of mechanical work once the patient reports to the hospital. However, it has been shown that the proton beam is capable of producing the flash effect, and it is meeting the biological mechanism of the oxygen level depletion and helping the sparing of the normal tissues. From the electron beam side, a very good theoretical work has been published by Nick Gaji et al. They have demonstrated here that the electron beams of energy 50 MeV to even 1200 MeV can be selected for producing the flash effect, but the lower is the energy will be the better approach for us. So ideally 100 to 150 MeV electron beams are thought to be a better candidate, and it has been further studied by other researchers. So now this is the one of the dosimetry work presented by Siddiqui et al. And this work was published in 2023. And from dosimetry point of view, if we look for the flash radiotherapy dosimetry, we look for, there are three important parameters. The dose rate linearity is the requirement of the dosimeter, which is having limitation size of now, a spatial resolution of the dosimeter, and temporal response. So a work has been carried out by James Shaw, and he has compared three dosimeters: chemical dosimeters, ionizing these ion chambers, and luminescent dosimeters. So chemical dosimeters, now you can see from this spider plot, this yellowish part is showing the characteristic of the luminescence dosimeters. Luminescent dosimeter contains TLD, OSLD, as well as the radiation dosimeters based on Cherenkov radiation. So here we see that it fulfills almost all the requirements. It can have the linearity up to the order of 10 to the power 5 gray per second. The temporal resolution is of the order of nanosecond, and the spatial resolution is also of the order of sub-millimeter. So probably it seems to be one of the best available dosimeter as of now. However, very recently a good work has been done in Italy. Marinelli has presented a work showing the diamond, CVD diamond, but produced in a different manner, that is flash diamond. And they have measured the dose rate from a low energy flash radiotherapy system, and they have reported that this dosimeter seems to be meeting the requirements of the dosimetry in flash beam. As far as the clinical theoretical work is concerned, treatment planning systems has been developed, and they have studied few cases for comparing with the different flash beam radiotherapy system. So you can see here, this is the case of glioblastoma. These are the axial dose distribution, and finally the dose volume histograms for 250 MeV proton therapy system, 2 MeV high energy electron therapy system, and 100 MeV high energy electron therapy system. If we analyze the axial dose distribution and even the dose volume histogram, hardly any difference is seen. So only difference we find here at the normal tissue complication probability. It seems somewhat much better in case of proton, as well as in high energy electron beams of either 200 MeV or 100 MeV. Similar work was repeated for esophagus. Here also once again we see that the axial dose distribution and the dose volume histograms are matching with almost each other with
sorry sir, Dr.
slight
Sharma sir,
variation. Yeah. And then finally for prostate cancer, it also seems to be almost comparable with each other. So in conclusion, flash radiotherapy appears to be a promising treatment technique for cancer patients and have the potential of enhancing the tumor control probability as well as minimizing the normal tissue complication probability. This technique has generated a lot of excitement in the radiation community over the past few years, and a number of reports has been published. A few compact, easy-to-use systems has been proposed. However, these systems are not yet available, but prototype systems has been made for some limited clinical trials. However, as a regulator's point of view and as from the radiation safety point of view, systematic clinical trials of all these systems are required before commissioning them in the practice. So thank you very much.
Thank you, Dr. Sharma. Very nicely covered the journey of the radiotherapy. Any questions from the audience? No.
Actually, there's one there here.
Yeah, very nice coverage of flash radiotherapy, which is one of the emerging topic in the radiotherapy. So there have been a lot of about mechanistic angle. As a radiation biologist, we are more interested about mechanistic angle, that when you give flash radiotherapy, and there is a very sudden drop in the oxygen sort of a consumption is too lot. So whether such type of study is also done in case of some in vivo clinical scenario to prove that whatever happens in the cell level, there's also in the patient level something? Is something known?
Actually, a few patients has been treated in Netherlands very recently, about 6 months back. Okay. So their follow-ups are going on. As far as the work which could be conducted in India, we are having a industrial accelerator which has the dose rate of the order of 5 kg per minute. So in fact, I was advising my group to use this system for in vitro studies. So probably they are initiating some work in EBC Kharghar, okay. Electron Beam Center.
Maybe we should start something together.
Yeah, yes.
Thank you.
Thank you.
Dr. Sharma sir, actually the most beneficiary in this whole exercise is our patient. For patient to come repeatedly for the hospital to get a treatment almost 30 to 35 times is really a problem for them. And the number of patients that we have, and to treat them, if it can be reduced to a flash radiotherapy within a one sitting, nothing like it, and they are the most beneficiary. The bugbear for radiation oncologists is always the hypooxygenated area in the tumor cells, and this flash radiotherapy can take care of it. And another point is regarding the tumor stem cells. And whatever the type of radiation currently that we are using it, they are not really touching the tumor stem cells at all. And this flash radiotherapy can take care of that, which will really, a later stage, will reduce the chances of recurrence of that tumor. With this, my comment, and we'll close this session. Thank you very much, Dr. Sharma Sahab, and also all the four speakers, and give an excellent talk on various topics on radiation research. Thank you so much. I thank my co-chairpersons also.
Thanks.
That was a wonderful session. With this, we conclude this session. Hope everyone is having a nice session. And now I call upon stage Dr. Amrita Rakesh and Anita Kumari to proceed with the inaugural function.